Welcome to the fascinating world of the genes with Rohini, and today we are going to learn about the genetic imprinting or the genomic imprinting. It is a topic from the eukaryotic gene expression. Now, what do you mean by the genetic imprinting or the genomic imprinting? Genomic imprinting is an epigenetic phenomena, okay, that causes that causes genes to be expressed in a parent of the origin specific manner. Now, what does this mean? Epigenetic phenomena, it is what is the epigenetic? It is the non-Mendelian inheritance. In that, there is a no change into the sequence of DNA. Still, the pattern of the expression of the genes from the parents to the offspring, it is going to be inherited. And for this inheritance, okay, what is used? The DNA methylation. It is used, and this DNA methylation, it is going to occur into the germ cells. And this DNA methylation in the germ cells, it is responsible for the phenomena called as the imprinting. And here, what does this describes? Okay, this describes the difference in the behavior. Okay, difference in the behavior between the alleles inherited from each parent. What do you mean this? Okay, to understand this line, let us take the example of the two genes, and it is the IGF2 and the H19. Okay, these are the two genes which are present over the same chromosome, chromosome number 11, and these genes, it has a single enhancer. That is, for the expression of the H19 and the IGF2, there is a need of the enhancer, a single enhancer, it is going to be used up. Okay, there is a placement of this ICR. ICR, it is imprinting control region. Okay, imprinting control region, or it is also called as the differentially methylated domain. Okay, differentially methylated domain. Okay, so in this, okay, in this ICR, there is a presence of the insulator. Okay, now you may know that the function of the insulator. Okay, insulator, it allows the binding of the repressor, and it is going to inhibit the effect of the enhancer over the next gene. Or this insulator, okay, this insulator, it is going to insulate the spreading of the methylation. That is, it is responsible to inhibit the effect of the expression, and also it is responsible to inhibit the effect of the silencing. It is like how in your lab you have the incubator, or you have the oven, and it has the insulator, and that insulator, it is not allowing the effect of the outer environment into the inner, and the effect of the inner environment to the outer environment. Okay, that is, it is going to insulate. Okay, it is going to do the it is going to insulate the conditions which are present into the hot air oven, or it which is present into the incubator. Okay, so in this way only these insulators, okay, these insulators, it is not allowing the effect of the enhancer over the genes, or it is not allowing these insulators, it is not allowing the effect of the spreading of the methylation over the expressing genes. Okay, so for, okay, for the, okay, for the imprinting, okay, for the imprinting, for the differentially expressed gene in a specific manner, what is required? There is a requirement of the methylation, there is a requirement of the insulators. Okay, so these are the two chromosomes. Okay, on the two chromosomes, the two alleles, they are present. Okay, two alleles, IGF2 from the maternal side, from paternal side, H19 from the maternal side, H19 for the from the paternal side. Okay, so the all genes, they are present into the 2n number. Why? Because the nature of the eukaryotes, it is a diploid, and as it is diploid, there is the two allelic forms, they are present. And here in this imprinting, the parent specific manner, there is the inheritance, means the one gene, it is only going to be expressed from the one chromosome, and the second gene, it is going to be expressed from the second chromosome. Here take the maternal chromosome. Here and the genes, they are the same one, IGF2 and the H19 gene. And here in the maternal chromosome, when you look at the expression, what you observe here, this H19, it is expressing. Here it has been written on means it is expressed one, it is expressing, that is the enhancer. It is allowing the binding of the activator, and that activator now it is going to act on the promoter of the H19. So there is an expression of the H19. But here what you observe in this diagram, this activator, this enhancer, it cannot act over the IGF2. Means the IGF2 it is silenced. Okay, IGF2 it is not expressed. Okay, in the from the paternal side of the chromosome. So here the expression pattern, it is understand the expression pattern which is decided here. It is how from the maternal side H19 it is only going to be expressed, IGF2 it is not going to be expressed. And why this is going to happen? To balance, okay, to balance the product from these two genes. There should not be the overproduction of the product from the genes. If the two genes they are going to be expressed, to n number of the product it is going to be synthesized, and that to n number of the product it is not required by the cells, it is not required by the organism. So the n number of the product only it is required. So the one of the gene it is going to be a silent one. So the expression of this IGF2 from one chromosome it is only required. Expression of the H19, only one gene product it is required. So one gene it is made silent and another gene it is allowed. So from the maternal side it is decided that the H19 it is going to be expressed and IGF2 it is getting silent one, but the IGF2 it is allowed to get expressed from the paternal side of the chromosome, and the H19 it is going to be inhibited, it is not expressed. Means when there has to be the expression of the IGF2, the enhancer it should act over this IGF2. Enhancer it is allowing the binding of the activator, and that activator now it is going to allow the expression of the IGF2. So from the paternal side of the chromosome, IGF2 it is going to be expressed, and from the maternal side of the chromosome, H19 it is going to be expressed. So this it is a pattern. Okay, this it is a pattern of the expression, and that pattern of the expression it is going to be inherited to the offspring. Okay, so here what is written here? The phenomena that causes genes to be expressed in the pattern of the origin specific manner. Means if it is a maternal chromosome, allow the expression of the H19. If it is a paternal side of the chromosome, allow the expression of the IGF2. This it is a parent of origin specific manner. And the two alleles, okay, two alleles they are behaving, okay, differentially. How? The IGF2 it is silent, okay, IGF2 is silent from the from maternal side of chromosome, and H19 it is a silent from the paternal side of the chromosome. Two alleles they are present, but the two alleles from the two chromosomes from the parental side it is going to behave there. Its behavior it is how its behavior it is a difference in the behavior between the alleles inherited from each parent. Okay, two alleles they are present, but not both of the alleles they are expressing. One it is silent and one it is expressed from each parental chromosome. Okay, so how it is going to happen? Now just we have seen that in this diagram there is a ICR. Okay, ICR it is what? Imprinting control region, or it is also called as a differentially methylated domain. And this ICR it is consisting of this insulator, and this insulator it is allowing the binding of the CTCF. So in the maternal chromosome to the insulator there is a binding of the CTCF, and as there is a binding of the CTCF, it is not allowing the enhancer to act on this IGF2. Means the expression, okay, the expression of the IGF2 it is inhibited. Okay, the IGF2 it is not allowed to get expressed. Okay, when to the insulator, there is a binding of the CTCF. So this CTCF, by binding with the insulator, not allowing the enhancer to act over the IGF-2, thus there is an expression of only H19. There is only expression of the H19. There is no expression of the IGF-2 from the maternal chromosome. Now next, what is going to happen here? In the paternal chromosome, how only the IGF-2 it is expressed and not the H19 it is expressed? When there is inhibition of the H19 expression, at that time what is used here? Methylation. This ME, it is a methyl group which is added over this DNA. So there is a methylation of the chromosome. Here the methylation it is done over the insulator and also in the promoter region of the H19. So what is going to happen when there is a methylation over the insulator? This CTCF, it cannot recognize the insulator. So as a CTCF, it is not going to recognize this insulator. What will happen? There is a no binding of the CTCF to the insulator. So it cannot inhibit the effect of the enhancer, the activators which are bound to the enhancer over the IGF-2, means the IGF-2 now it is expressed. Okay, how? Not allowing the CTCF to get bound with the insulator. How? By the methylation of the insulator. So the IGF-2 it is expressed. And how? No activation of the H19. There is a methylation of the promoter, the region of the H19 it is methylated. So due to the methylation, activator cannot activate the H19 transcription. The activator it cannot equip the basal transcription apparatus over this H19 gene, and as there is a no RNA polymerase 2, okay, there is a no transcription of the H19. Thus the H19 it is off, that is H19 expression it is inhibited, H19 it is not expressed. Okay, so in this way the methylation, methylation of the DNA, it is allowing the imprinting. Okay, that is the stamping of the genes. It is allowed the imprinting, genomic imprinting. It is due to what? Due to this methylation and also the insulator which is in between this H19 and the IGF-2. Okay, so when there is a imprinting of the genes, there is a role of the insulator as well as there is a role of the methylation of the DNA. Okay, so there is a differential expression of the genes from the paternal and maternal side of the chromosomes, and this pattern it is going to be inherited to the offspring, and this it is what? This it is epigenetic, and for this purpose to have this pattern of the expression from the parent to the offspring, when it has to be there is a need of what? There is a need of the imprinting. When this imprinting it is going to happen? When the cells they are the germ cells. In the germ cells, what is happening? The methylation pattern, okay, or these two chromosomes which are methylated, the completely the methylation, methylation it is removed. That is, there is a demethylation of the both of the chromosomes in the germ cells. When it is a parent, when you look at the parental chromosomes, you observe that in the parents also there is a paternal side of chromosome, there is a maternal side of the chromosome, and that chromosomes they are differentially methylated. Okay, so when there has to be to exhibit the methylation pattern, at that time there has to be the erasing of the complete methylation over the chromosome, and that erasing of the methylation it is done into the germ cells, and the parent specific pattern, okay, if it is going to give rise to the oocyte, then the maternal type of the expression pattern it is going to be there. That is here, okay, complete, there is a complete demethylation, okay, complete removal of the expression pattern over both of the chromosomes. And if it is a maternal chromosome, or if it is a if it is a maternal chromosome, then at that time there will be the CTCF, it is going to be there, okay, on the insulator. And the in the germ cells, okay, in the germ cells, the all the chromosomes, okay, all the chromosomes, it will have the active form of the H19 and the silent form of the IGF2. That is, after meiosis, the four cells which are going to be formed, in the four cells, whatever the haploid nature it is present, it is consisting of the H19 as the active gene and IGF2 as an repressed gene or IGF2 as an silent gene. But when it is in the paternal chromosome, okay, in the germ cells, all the methylation pattern, all the expression pattern, it is going to be erased, and the new pattern, it is going to be executed. And if it is a paternal chromosome, then here in this case there will be the methylation of the insulator, and there will be the methylation here in the H19 region, so that the IGF2, it is only going to be expressed. So this methylation pattern or this expression pattern, where it is set into the germ cells. And then when there is a reunion of the two gametes, then you are going to observe that the both of the chromosomes, they are present, but the alleles, they are differentially expressing into the two chromosomes. Okay, so in this way there is a imprinting, and if there is a defect into the imprinting, then what is going to happen? There will be the occurring of the disorders. Now, just now we have seen that IGF2, it is going to be expressed from the paternal side of the chromosome, and if there is a mutation, there is a disruption into this IGF2 gene, then there will be the arising of the disorder, and here it is a Beckwith-Wiedemann syndrome. Okay, though here there is the non-mutated form of the IGF2, it is present to the maternal chromosome, still you are going to observe that there is arising of this syndrome. Means the expression pattern which has been decided, it has to be carried out. Though there is a defect into that gene, the maternal side of the chromosome or the paternal side of the chromosome, it is not going to do anything. Okay, the expression pattern which is going to be decided, it has to be executed by the cells. Though here in this case, here it is a mutation into the IGF2, though maternal side of the IGF2, it is good in condition, it is a functional one, still that IGF2, it is not going to be activated, and here you are going to observe that there is a syndrome. And here in this example you can see that, okay, you can see that there is a Prader-Willi syndrome and the Angelman syndrome. These both of the syndromes, they are occurring due to the deletions into the due to the 15q11q13 region of the chromosome number 15. You can see here, okay, the region, it is the same one. Okay, region, it is the same from the same chromosome, but the two disorders, they are the different one. How? If it is a paternally inherited, then there is a Prader-Willi syndrome, that is a PWS. Okay, and if the mutations, deletions into the region from the maternally inherited alleles, if it is there, then there is an Angelman syndrome. Okay, so here, okay, so here the genes which are expressing from the paternal side or the maternal side, if there are the defect, there is a arising of the syndrome. Though the regions of the paternal and the maternal chromosomes, they are the functional one, still you are going to observe that if here in this case paternally inherited alleles, if they are disturbed one, then there is a Prader-Willi syndrome. And if the maternally inherited alleles, it has the deletions or it has the mutations, then the Angelman syndrome, it is going to occur. Okay, so here if there is a defect into the imprinting, okay, if there is a defect into the imprinting, at that time there is the occurring of the syndromes, the disorders, they are going to be there. I think you may have cleared the idea about the genetic imprinting. If you all like my video, please subscribe my channel and also share my videos with your friends so the knowledge it is going to spread. Thank you.